Hi, everyone. Welcome to Naturally Immune for Life. Thank you for joining me today. Thank you for your time. I thought I would flesh out the microbiota is critical in immune disorders. So I thought I would follow up on this source from that and flesh out the um, gut microbiota in metabolic disorders for you. It seemed kind of stupid to end with such a grand finale and then not explain it a little in more detail for you. So that's what this post is going to be. So it is the gut microbiota in metabolic diseases. So the human gastrointestinal tract is home to a complex ecosystem of microorganisms known as the gut microbiome. This community includes bacteria, viruses, fungi, and other microbes that coexist with the human host in a symbiotic relationship. The gut microbiome contains a gene pool that far exceeds that of the human genome, accentuating its immense biological potential. Over the past 2 decades, advances in technologies have transformed understanding of the microbiome and its role in health and disease. Once thought to function primarily in digestion, the gut microbiome is now recognized as a critical regulator of metabolic processes, immune function, and systemic inflammation. These discoveries have led to increasing interest in the microbiome's role in metabolic diseases, particularly obesity and type 2 diabetes. Metabolic diseases are characterized by disruptions in the body's ability to regulate energy balance, glucose metabolism, and lipid storage. Increasing evidence reveals that alteration in the composition and function of the gut microbiome may contribute to the development and progression of these conditions. The composition of the gut microbiome is dynamic and influenced by a combination of host-specific and environmental factors. Genetic background plays a role in shaping the microbiome, although its influence is modest compared to environmental inputs. The immune system is another determinant. It maintains an intricate balance by tolerating beneficial microbes while limiting the growth of harmful organisms. Disruptions in immune regulation can lead to microbial imbalance, known as dysbiosis. Age is also a critical factor. The microbiome develops rapidly during infancy, stabilizes during adulthood, and declines in diversity with aging. These shifts have important implications for metabolic and immune health. So, among external influences, diet is the most significant modulator of the gut microbiome, including the infant diet. Long-term dietary patterns can shape microbial communities. Diets rich in fiber promote the proliferation of bacteria that produce beneficial metabolites, while diets high in certain fats, and refined sugars are often associated with reduced microbial diversity. Antibiotic use represents another major disruptor, leading to long-term alterations in microbiome composition. Other factors, and another thing is that infants don't eat fiber, so that only comes into play in toddlerhood. Other factors such as physical activity, stress, and exposure to environmental toxins may also influence microbial diversity and function, although these relationships are not well understood. The gut microbiome and obesity. Obesity is one of the most extensively studied conditions in microbiome research. Early studies identified differences in the relative abundance of major bacterial groups in lean versus obese individuals, revealing that the microbial composition may influence energy balance. One proposed mechanism involves the microbe's capacity to extract energy from the diet. Certain microbial communities are more efficient at breaking down complex carbohydrates into absorbable compounds, thereby increasing availability. This enhanced energy harvest may contribute to weight gain over time. In addition to energy extraction, the microbiome can influence host metabolism through signaling pathways that regulate fat storage. The gut microbiome has also been implicated in the regulation of appetite and satiety. Through interactions with the gut-brain axis, microbial signals can influence hunger hormones and neural pathways involved in food intake. And the first one is probably not it because the immature microbiome cannot extract energy efficiently. And this particular resource is from 2013, and so they later went on to pretty much eliminate that. In addition, that the breaking down complex carbohydrates into absorbable compounds, well, that was later found that it's not carbs, it's actually sugar that breaks down to fructose and glucose. So they eliminated the sugar and kept the carbs, and then they were able to extract energy more efficiently, and have more energy, even though they were eating high carb junk food. So type 2 diabetes is characterized by impaired, and in that particular instance, it was the fructose. So type 2 diabetes is characterized by impaired glucose metabolism and insulin resistance. Increasing evidence suggests that the gut microbiome plays a role in both the onset and progression of this condition. Individuals with type 2 diabetes commonly exhibit reduced microbial diversity and altered functional profiles. These changes may affect the production of metabolites that influence insulin sensitivity and glucose homeostasis. An important mechanism involves inflammation. Chronic inflammation is a well-established contributor to insulin resistance. Type 1 diabetes. Even though the autoimmune disease type 1 diabetes has a known genetic risk factor involving mutation in the human leukocyte antigen genes, the recent rise in the incidence of this disease points to environmental factors playing an increasing role. A study of 4 children with newly developed type 1 diabetes and 4 matched control children found differences in the composition of their gut metagenome between the groups and reduced diversity in type 1 diabetes associated metagenomes. Studies in non-obese diabetic mice have shown that germ-free non-obese diabetic mice, or those housed in specific pathogen-free conditions, are more likely to develop diabetes, suggesting that the gut microbiota are involved in the development of autoimmune diabetes. Specifically, a particular species of bacteria was found to protect against autoimmune diabetes in non-obese diabetic mice. Interestingly, children who progress to develop type 1 diabetes have a markedly altered serum metabolome that could already be detected in the cord blood. So the gut microbiome exerts influence on host physiology through a range of functional activities mediated by microbial metabolites. One of the most significant contributions of the microbiome is the production of short chain fatty acids, including acetate, propionate, and butyrate. These compounds are generated through the fermentation of dietary fiber and have beneficial effects. Short chain fatty acids serve as an energy source for intestinal cells, strengthen the gut barrier, and modulate immune responses. They also play a role in regulating glucose and lipid metabolism. Immune system modulation. The microbiome is integrated with the host immune system. It helps educate immune cells, promoting tolerance to beneficial microbes while maintaining the ability to respond to pathogens. Disruptions in microbial balance can lead to immune dysregulation and chronic inflammation. This inflammatory state is a major factor in the development of metabolic disorders. So when it comes down to it, that is the most likely factor. Host microbe communication, and we know this from 2016 about milk, a crucial postnatal signaling, and crucial postnatal signaling and the regulation of the immune system and homeostasis. Host microbe communication. Communication between the microbiome and the host occurs through multiple pathways, including hormonal, neural, and immune signaling. The gut-brain axis is a prime example, linking microbial activity to the central nervous system function. Through these communication networks, the microbiome can influence not only metabolism, but also behavior, mood, and cognitive processes. Studying the microbiome, advances in technology have been essential for uncovering the complexity of the gut microbiome. Next generation sequencing techniques allow researchers to analyze microbial DNA at an unprecedented scale. The vast amount of data generated by sequencing requires sophisticated computational tools. Bioinformatics plays an important role in analyzing and interpreting microbiome data. These approaches help identify potential causal relationships between microbial features and disease states. Animal models, especially germ-free mice, are widely used to study the microbiome. And in the last post, you see that's being abandoned somewhat. These models allow researchers to isolate specific variables and investigate causal mechanisms, and they actually haven't been shown to do what they hoped they would do, as we covered the last time. For example, transferring microbiota from obese individuals to germ-free mice can result in increased fat accumulation, providing evidence for a direct role of the microbiome in obesity. Despite significant progress, microbiome research faces several challenges. One major issue is distinguishing correlation from causation. While many studies have identified associations between microbial patterns and disease, proving that these microbes directly cause disease is difficult. And we also went over that the last time. It's not only difficult, it's impossible. because you can't actually have good controls. Individual variability and the sheer number of microbes makes it impossible. Individual variability is another challenge. A person's microbiome is unique, influenced by a lifetime of environmental exposures and other lifestyle factors. This variability complicates the development of universal treatments. The microbiome is dynamic. Short-term changes in diet, environment can immediately alter microbial compositions, making it difficult to draw conclusions. That's true because that alone can change your microbes. And however, that doesn't deny the fact that there can be effective universal treatments because the infant colonization of bifido is universal. The field of microbiome research is evolving at a fast pace with promising implications, and there are also other universal microbes that the majority of people have. And the environment, the diet doesn't really play a factor until the adult microbiome is beginning to be formed. The field of microbiome research is evolving at a fast pace with promising implications for clinical practice. Personalized medicine is one promising prospect. By analyzing an individual's microbiome, clinicians may be able to design targeted interventions to improve metabolic health. Therapeutic strategies aimed at modifying the microbiome are also being investigated. These include probiotics, prebiotics, dietary interventions, and fecal microbiota transplantation. And as we covered last time, FMTs are probably where it will end up. As understanding develops, the microbiome may become a central component of strategies for preventing and treating metabolic diseases. And this is also including atherosclerosis, which I did not include that section in here, although you can find it in the source. The gut microbiome represents a complex, influential component of human biology. Its role extends far beyond digestion, encompassing critical functions in metabolism, immune regulation, and systemic health. Sorry for that noise. The wind is making my door open and close. Evidence increased, even though I put a doorstop in front of it. Evidence increasingly supports a link between alterations in the microbiome and metabolic diseases such as obesity, atherosclerosis, and diabetes. So as I said, I didn't include the atherosclerosis section, but it is in the source. And it was also covered in the last post, which was microbiota plays a crucial role in immune system disorders. Ongoing research continues to shed light on the mechanisms underlying these relationships. Ultimately, a more comprehensive understanding may lead to innovative approaches for managing metabolic disease and improving overall health. So, as I said, that is from 2013, and there have been quite a few advances since then. But this is how long they have been actually looking at this, and I'm sure there are probably earlier ones as well that I just haven't seen. New research on human milk sugars, human milk oleosaccharides help us understand how these prebiotics support a healthy gut and strong immunity starting from birth. Over 200 HMOs have been discovered so far. HMO supplements not derived from human breast milk have recently been developed. And these are also being included in some infant formulas. So, I recently saw these new products, so I have put some of them in favorite products in case you're interested in checking them out. I had only known of one up until this point, and that was Holy Ghost, and it was actually pretty expensive. And I have the chart here for one, and it is supports brain health, 5 key types of HMOs, boosts the immune system, promotes the growth of probiotics. I think it says 200 billion in one of them. Supports gut diversity, boosts cognitive function. It also helps form tight junctions. So there are single ones from this particular brand called Layer Origin. There are single ones, but why get the single one when you can get 5 in one, even though I have them all? So you can check those out and see which they are all numbered by HMO. Some have HMO2, some have HMO3. But as I said, why do that when you can get all 5 and get the benefits that they claim in one product? They are a little bit expensive, but they do have a kids formula that isn't too bad. And it also has 5 HMOs. And I also want to talk about something else they found here. Let me go to my favorite products page. And this was a probiotic, prebiotic that specifically, okay, there's also a pure bifido with 5 strains of bifido. And it also has an HMO prebiotic. And then there is another one called Longevity. And it also has prebiotics that specifically claim to detox glyphosate. So I know some people are particularly interested in that. So those are just some of the newest things I've run across that in reading the comments or the reviews, I mean, people seem to really like them, and that might be something you want to check out. So the particular one for gut barrier function was HMO3. And just let me go here. Interestingly enough, I'm trying to think about what happens in GCMA when you do not have enough GC protein. So let me get this chart up here and see if I can remember my science here. So this particular one is Sialac, and it is about sialidase, which is an enzyme that you are also supposed to be producing, along with galactosidase. So let me see if I can make this larger. Okay, so a unique HMO that works from inside to restore, protect, and soothe your digestive system, fortifies the intestinal barrier, reducing leakiness and promoting structural integrity, stimulates the regeneration of cells for a stronger and more adaptive digestive tract. Let me see if I can make this bigger. Of course I can't. Resilient. Can't. Microbiome balancing, since selectively nourishes beneficial bacteria. Sialactase. Actually, I was looking for sialidase. So anyway, it has a little chart here showing you how it restores tight barrier function. And that is from HMO. So I think you might find that interesting regarding leaky gut. And, oh crap, I meant to look at the exact price of that for you. So it's $32 for one pack. I don't know what the 60 tablets, one per day. It's not too bad if you're interested about leaky gut, but as I said, why just get the one when you can get 5? So this one is more expensive at $70. And they also have it in capsules, which is less expensive. I don't know why I can't see that. There's a mistake in that link. So anyway, let me just show you the other first one I found. I think it was $32 for 45 servings. So you might want to check those out, and I will definitely fix that link because I don't know why it's not working, but so that's welcome news. They're also beginning to put these in some infant formulas. Which ones I don't know, but you can look particularly for HMO, but remember it does not in any way replace breastfeeding because, as I said, so far they have found over 200 HMOs, and it's going to take them eons to discover what they do, and they couldn't actually see these HMOs until about 20 years ago. So isolating them, seeing what they do, how they function. I mean, some of them might work similarly. I don't know, but I know this is emerging science, and you will not get to the end of that ever, if ever, because I'm quite sure there is well over they've already discovered over 200, and they keep finding them. So, okay, everyone, that's it for this time, and I thank you so much for your time. If you're interested in those products, they are in my favorite products, and it might be something you want to add, and that might help shorten the time frame of just using colostrum yogurt, kefir, and whole fat mammal milk. That I don't actually know, although it seems from the reviews, many people notice a difference almost immediately. So that just goes on to prove what I've been saying all along, that you cannot replace mom. Okay, I'll see you next time, and thanks again for your time, and you have a great day and a great week. Bye.